Hello and welcome to this science talk today where we're going to talk about fast-tracking vaccine development for the COVID-19 pandemic. I'd like to warmly welcome our three speakers. First, Translational Søgaard, you're the Vice President at Biotechnologies. Thank you for coming. Salanti, founder and board member at AdaptVac and also professor at Copenhagen University. Thanks for coming. And Louise Hefting, you are a field application scientist at ChemoMetec AS. Welcome to you too. So we're going to talk today about vaccine development for the COVID-19 pandemic. And under the Prevent and COVID consortium, which is a collaboration between several academic institutions and the biotechnology companies, you, Ali, and Max have been involved in that. In Denmark, the consortium members are Copenhagen University, AdaptVac and ExpreS²ion. We're going to learn how you've worked together on that. Louise, you are here to talk to us a little bit about how a company like ChemoMetec support a consortium like the Prevent and COVID to succeed at their goals. So first, Max and Ali, I'd like to talk a little bit about your technologies and then we'll move into some of the experiences on fast-tracking this vaccine development. So Ali, could you tell us a bit about AdaptVac and your VLP technology, which is the virus-like particle platform that you have developed?
Yes, it actually goes many years back and allow me to start that. So we've been working with vaccine development at the university for 25 years, aiming at making malaria vaccines, but also other types of vaccines. So we followed the vaccine field very closely. And what we have seen with other vaccines, and including the malaria vaccines that we have also brought to the field, is that they simply fail in inducing a very strong immune response. And that has become now more or less the consensus with most vaccines being tested with all these new technologies available. All the old vaccines, they still work against all the diseases that we have almost eradicated, but all the new vaccines emerging, they do simply not work. So we sat down at one point some years ago and said, what is wrong? Why don't vaccines work as we had aimed at and hoped for, and started to look at what are the different vaccines out there that actually work and what are the ones that do not give this response that we are aiming at. And the one that stood out was the HPV vaccine. The HPV vaccine is a remarkable example of a vaccine giving lifelong protection after 1 or 2 vaccinations. It is a recombinant vaccine, so it's made with these modern technologies. And what we learned was that this vaccine is so effective because it is a virus-like particle. It has a very dense display of epitopes, and this is what triggers the long-lasting plasma cells that gives this memory response and keep producing IgG against the HPV particle. And that's also why the soluble vaccines, they fail in doing this, because they don't have this repetitive surface. So they don't give this memory response. You see strong peak responses, you get a fine response, but they fade out. So we decided to try to develop a platform where we could make any vaccine, any target, could be malaria, it could be influenza, look like a HPV particle. So we took together at university, we invented this technology with super glue, enabling gluing vaccine targets like the COVID spike protein onto the surface of an empty virus capsid construct. So it looks like the HPV. It has the same size. It has the same repetitive, dense display on the surface enabled by the superglue. And then we started vaccinating against different targets. And what we found was that we get a very strong response, and it's a long-lasting response. We demonstrated that with a cancer vaccine and with an influenza vaccine. So we filed patent applications on the technology and founded AdaptVac to move this forward to clinical trials.
Thank you. And Max, maybe you can share a little about what ExpreS² Biotechnologies does and your ExpreS technology.
Mm-hmm. So the ExpreS technology is a platform which was spun out from a company called Pharmexa, by the people at Pharmexa that developed the platform. And so the difference to other platforms is the cell line, which is a cell line based on fruit flies. And so basically, a screening project was undertaken, and it was found that these cells are actually really good at both R&D development, but also transferring that to production due to the qualities that they have. And over the years, we've shown that they are in fact able to make proteins that fail for one reason or the other in other systems, and malarial proteins is a good example of those, where in some instances a few milligrams were maybe seen on a gel, micrograms were seen on a gel at some point, and then we can make hundreds-fold more. And so with the protein mechanics in this project, no. Now COVID isn't a difficult protein to make, the spike protein, and many other platforms can make it. But in this project, we've been the quickest to make different variants, and we could, in collaboration with AdaptVac, quickly screen through a lot of different constructs to arrive at the lead candidate. So that's been our role here.
Thank you. And why did the two of your two companies partner up for this project?
Yeah, so we had a long-lasting collaboration on malaria vaccines because the insect cells, maybe because of the life cycle with the malaria parasites also having a part of its life cycle in insect cells, we don't know, but they are particularly good at making malaria proteins. So we've been making malaria vaccines together and taking that through clinical development. So it was obvious to work on this project as well.
Mm-hmm.
Thanks. And maybe to understand your infrastructure a bit, Max, could you briefly just introduce how you as a consortium worked with industry partners, AGC Biologics and Bavarian Nordic?
So to begin with, Bavarian Nordic wasn't a part of the consortium, of course. That agreement was made later on. The construction was in fact the brainchild of my boss, Vian de Jong, who is the CEO of AdaptVac as well. He had, from the malarial projects, he had the network to both production at AGC, to clinicians that he knew, and to regulatory authorities, so that he could quickly sketch out a pathway to go from a gel to phase 1. And had he not written a brilliant application together with Morten, I believe, from AdaptVac, this wouldn't have happened, basically. And so he's the real hero in the story. And so basically AdaptVac is heading this. We have engaged industrial partners. They have engaged industrial partners, and that's how it works. And because AdaptVac and ExpreS²ion, we know each other really well, and so there's been a very fluid collaboration, both with protein expression, process development, and so forth.
Which has probably also been necessary because of the fast tracking aspect of it. Yeah. So, Ali, what is the status currently on your vaccine?
So, we've been working very hard on getting this into a product that can go into humans, and it's not trivial. There's a lot of different phases working with this new technology, the virus and the super glue and the protein that we need to glue onto the surface. So we had to work with processes at a speed that enabled to meet the requirements to go rapidly into humans, but we didn't have much time for process development. So much of what we have been doing has been at a certain risk. So we are extremely relieved that we're now done with the GMP production, and it's being vialed now, and we're starting to recruit the first patients.
Congratulations. I mean, that is great news. So you mentioned a bit about it being risky, and with the eyes of the world almost looking at you and the other people doing some of these vaccines for corona, you must have felt at least some pressure. So what have been some of your considerations before you decided to go into this work?
So for a modest-sized company like ours, the consideration would be that we spend 100% of our time doing this, and so it's really been all hands on deck, and maybe helped by a downturn in the customer business due to COVID. It was less disruptive, but that has been really putting everything on at stake to just do it, and also depending on all the other partners. So normally in vaccine development, life happens and things take longer, and so this project has been reliant upon both us, AdaptVac, AGC, who makes it as well, to think outside the box, because and be on the ball. Otherwise, this wouldn't really have happened. And there's been many points along the way where everything could have failed, but it didn't, because someone stayed up all night. I think, yeah.
And I think from our point of view, at least from the university, it was obvious that we should do this, because we had the technology, and we do strongly believe that this can be a really, really good vaccine. Just before COVID hit, as a pandemic, we were finalizing a flu vaccine, and we just published that on the same platform, showing really nice protection in animals. So it was obvious for us to get started on this, and we got the EU grant, obviously, as we have heard, but also support from the Danish Research Councils, and not the least Carlsberg Foundation and another Danish foundation, Honoré. And with their support, that enabled us to gather a larger team than what we had in the company, where we only a few people employed. So we were allowed to work at the university facilities because it was closed down anyway. So as an academic project, it moved forward in an otherwise empty building during the lockdown. So for us, it was a normal workday, because we used to working with diseases and making therapies and vaccines against serious diseases like malaria and also cancer. So there's always a pressure on delivering. So in that sense, it wasn't that different. But obviously, clearly the timelines, because of the compression of the timelines, then there's been an added workload and added pressure on the people doing the work in the laboratories.
Yes, obviously. So actually, I'd like to focus a bit on this, the fast tracking and how to move through that. So could you maybe say or explain a bit about how you did this vaccine development differently than your normal pipeline of development, and what were some of the shortcuts or the bottlenecks that you could maybe skip or move through faster, or however you did this? So maybe Max, or maybe Ali first. Sorry, because
so I mentioned before that we worked at risk. So doing a GMP production, transferring everything to the production plant, the CMO, is a big thing. And when they get started, you cannot do changes. Then it's all or nothing. And you book a time slot at the production, and it costs around those 2 million euros. So it's not something you can do in an iterative process. So for the malaria vaccine that we did together, and also for other products that we have made, then you have a very long period ensuring that everything is fine, and that this process here cannot fail, even though it fluctuates a few temperatures, or the pH a little bit is changed. So we didn't have time to that. We made the process, and then we transferred it to the CMO, and then we crossed the fingers that at the end we got a viable product. So that differs a little bit from how we would normally do it. It doesn't and it's important to say, it doesn't compromise the safety, because it's the same requirements for releasing the product. It should be free of virus and DNA and endotoxins and all that. So that is not compromised, and it's the CMO who does the analysis. And if it meets the requirement, then it's fine. And if it doesn't, then we wasted our time.
So it's more of that iterative process of going back and forth and optimizing your system that you didn't find the time necessarily to do, and or at least not as much, and you were maybe even a bit lucky in getting it right the first time.
Yeah, and maybe I could add that this has maybe also learned us that we should maybe work in other projects at a little higher risk to move it faster.
Yeah.
To be a bit more daring.
Interesting. And Max.
So also we have employed an affinity tag, which has been clinically validated in a different malarial project, and so that is a thing that makes the downstream, the purification of the protein much simpler, because you can just skip a lot of other steps that you would have otherwise have to do. And so and also we've employed, so we normally make cell lines, and these cell lines are basically producing the protein of interest constitutively, secreting it to the cell surminatant. It takes 3 to 4 weeks to select such a line, and so 3 to 4 weeks is not really compatible with screening a lot of different variants. So we've done that by large-scale transfections, just purified the protein, sent them off to university for checking that they work, and so that has been something instrumental in keeping constructs and proteins coming. And then the affinity tag is basically transferable because it has been clinically accepted in a phase 1 study. That really makes the whole development process much faster. And then for the transfer, as I said, we had no time for technical runs. So there were knowns, so there were known unknowns, and there were also unknown unknowns, and so that kept some of us up quite a lot.
A few sleepless nights,
Yeah.
and what, hoping that it would just pass through tech transfer, or also, I guess, supporting that.
Supporting that, and but I have to say that this also on the AGC side has been mirrored by a lot of involvement and above the call of duty to make this work. And I think the urgency of COVID is what spurs people on, and that's actually quite important, and not something you will always have unless it's another pandemic.
Yeah. And if I can add to that, because it's absolutely correct. People have done more than what we could expect of them. So we have had companies reaching out saying, we've seen in the news you're developing this. We have expertise in downstream process analysis development. There was some of my former colleagues working in Måløv in Novo Nordisk who said, maybe we can provide this analysis for you. And yes, please. And you guys came and said, you need this cell counter. Can we provide it? And yes, please. And you came immediately and put it up. And the same with we have a biosensor. Atana came out and installed it, and Merck came out and helped with filters. So we have experienced a lot of goodwill and support, which under normal circumstances, would take time, and you would have to pay for it, and you would have to apply for the money. So that has also enabled fast track.
Yeah, because that sounds like a really amazing situation to be in, which is not the norm. So is that, or any other, is that your best experience, and also some of the advice that you would maybe give to others who are thinking about going into vaccine development for coronavirus or in general? Max?
So the good advice, I think advice is know your people, and do not take for granted that everything will go to plan. And if your project makes sense, people are actually up for helping a lot more than you would think. And in filtration, PAL has helped enormously as well. So, I mean, really, as long as people, we're all actually scientists in this, and our prime motivation is to make sense of what we do, even if we work in a company. And so if people have the chance to actually make sense or make a job which makes sense, they will do so and above what they're paid for. Right.
Yeah, I think and I hope that on the other side of this pandemic, this has spurred some public-private product development partnerships, and that will survive this, and that it will enable us, both in the company but also at the university, to develop new products with the contacts and the support we have from the industry.
So thank you very much for introducing this exciting journey of yours. Now, Louise, you come from more of a industry partnership point of view. Can you maybe tell a little bit about how you supported AdaptVac and ExpreS² in this vaccine development?
Yes, for sure. So we at ChemoMetec, and as long as the whole world were following the corona pandemic, and the need for developing a vaccine very quickly is apparent, a very, very important step. So we agreed to look into how we could support and help in this situation. And back in March, we saw the announcement that the consortium in COV was established, including the ExpreS² and AdaptVac. And in that relation, we agreed to contact Ali, and we did so, and we had a short discussion on, hey, can you use automated cell counting in your process? And you were really willing and arguing, yeah, let's try it out and see. And we tried to act as quickly as possible and went to the University of Copenhagen and had a nucleocounter placed in your lab.
And as a technology provider, what are the challenges you see typically in vaccine development?
Yes, so I'm not an expert in vaccine development, but somewhat in cell counting. But still, the general, I would say, in a development process is, as you also point out, the transfer between the different parties in a pipeline. Normally, we see an R&D situation develop PD process development, and then manufacturing and quality control. Sometimes it's the company has the whole pipeline, but often you actually see that you have to collaborate, biotech companies and the R&D structure, which is also the case here. So, I mean, if you do not have the same instruments and technical equipment available in the different steps, it's a challenge, and you risk to use a lot of time in the validation process of this.
And your business is cell counting. So what are the challenges in cell counting specifically?
Yes, actually, cell counting is not that easy as you might think. There is no true value of a cell count. Different counting principles will give you different results. So you need to stick to one in order to have the same process going through your pipeline. I would say a good advice from my end is to look into the ISO guidelines, which will give you some requirements on how to validate your cell counting process to address precision and accuracy.
Thank you. So Max, did you actually experience any problems in cell counting?
There were problems in cell counting. There's always problems in cell counting, especially if you have some cells that are not the run-of-the-mill cells like CHO. These are a little bit smaller, they have the big rounder, and just more fun altogether. But anyway, I mean, so there's always this thing about do you have the right number? Because if you don't, and you seed out these cells at the wrong density, then the cells will tell you because they won't grow or they'll overgrow. And so this is always a challenge. Yeah.
And Ali, what is the benefit to you of having industrial partnerships such as the one with ChemoMetec?
Yeah, so I mean, We are not GlaxoSmithKline or Novo Nordisk, so we are limited to the few resources we have and the infrastructure available. And we are always dependent on primarily grants and small investments. So it's not when we need something, we just can't go out and just buy it. So we are quite dependent on people like you donating or showing up, helping us with the processes. So it's really important, and we hope that it can be a long-term relationship with many of these companies, and that we also can maybe help develop applications.
Yeah, thank you. So if we look at COVID-19 vaccine generation in general and the fast tracking, is it too late to jump onto that train if a new company wants to start out in it? Ellie?
Well, from the beginning we knew that we would be very late, and that was not a problem because there are no diseases where you have a vaccine or a cure within 1 year. It's always an iterative process, and you need to learn how one treatment affects the disease and whether it can protect and to what degree it can protect. What we have seen is all the fastest vaccines, which is the live attenuated coming out from China. It's the adenovirus coming out from Oxford and Johnson & Johnson, and it's the DNA vaccines and the RNA vaccines from Pfizer and Moderna, BioNTech and CureVac. So they're very rapid to make, and we could also have, we don't have the IP, but we could easily have made such a vaccine in also a short time. We just don't really think that they might give the high level of long-term protection, as I discussed before, because they give it as a soluble protein. Even though it's an RNA, it's translated into a soluble protein. So I think there is a high need for new types of vaccines, new kind of therapies coming out in the tail of the first generation vaccines. And I think a combination of different therapeutic strategies, maybe some fast vaccines giving acute protection, followed up by a booster with some better vaccines giving longer term protection. So there's plenty of room for development of therapies against this disease as well.
Yes, and I guess also vaccines or viruses, tend to mutate and evolve. So I guess also there's. Are you planning on keeping to develop new vaccines from your platform?
That's the plan. So the immediate plan is to make a breast cancer vaccine, so not a viral one. But that is because the system is strong enough to break self-tolerance against a human, we hope, tumor suppressor. It works at least in mice. And so we are very much into developing new things as a pipeline business, yes.
Thank you. So, Ellie, you briefly touched upon it, but the nucleic acid vaccines have really been of high focus for the coronavirus because they have been able to bring the vaccines into clinical trials at an unprecedented speed. Like, are this type of vaccines winning the race, or is your VLP platform, will that prove to be the better one in comparison?
I mean, we try to not talk about it as a race. Maybe because we are at the very end. No, it's obviously there first, and we have seen from the clinical data and also from the monkey data and mice data that they do confer protection, and that's really nice to see. So I think they definitely have a place, these vaccines. But when we look at previously data published from these vaccines, there's only a few published papers out there, but there is one from CureVac on a rabies vaccine, and there's one from Moderna on a flu vaccine. What we can see is that they can induce these functional responses, as we have also seen with COVID-19, but they disappear quite rapidly. After a few months, they're gone. I would expect and fear we see the same with the mRNA vaccines against this target here. It doesn't mean that you shouldn't go out and protect maybe healthcare people with these, but as a vaccine strategy for protecting the world and you and me, young people, against this disease, I don't see the RNA vaccines as being the solution.
And Max, what are the differences in generating like the protein production for your vaccine versus the production of a nucleic acid-based vaccine?
Those are completely different disciplines, basically. And I wouldn't, I'm not an expert on producing mRNA vaccines at all. The challenge with mRNA is stability. Stability is key because there's RNA splitting enzymes everywhere. The challenge with a protein-based vaccine is that it takes some time because you need to show safety, of course, and you need to make the cell line, you need to make the purification, whereas in the DNA and RNA-based field, they just need to change an insert, and all the rest of it is templated and the same. So protein vaccines will never be as fast, but in conjunction with a virus-like particle, they are a very efficient, I think, tool, and could also be within a little bit later than the other ones, a emerging disease tool as well because of the adaptability of the VLP.
So now I'm getting a bit curious. What are some of the weaknesses or challenges with your platform?
Speed compared is a weakness. That would be that you actually need to engage a lot of people to make it. If you don't have your own production, that's something that could be changed. If you don't have your own production facility, then you're going to rely on other people to help you, and that's something would be nice to have all in-house.
The main, you can call it a weakness, is that we haven't seen it work in humans yet. So it's based on a lot of immunological insight from the university and a long development process also in animals and monkeys. But we need the final proof that everything we believe and have shown in animals is transferred to humans. So we are looking so much forward to seeing this first vaccine and based on this platform here going into humans.
But then I guess it must be encouraging to see the data from the Oxford's AstraZeneca vaccine trials, because that technology is somewhat similar to yours, as in it's not nucleic acid based.
Well, it's not that similar to ours, in fact.
No.
Because they use adenovirus to infect cells to express the protein of interest. But the protein of interest is basically not presented in the way that the virus-like particle method does, and that's the whole key to raising this huge immune response that is also have longevity to it. Really, the DNA and RNA vaccines are a completely different child than what we're doing.
Yeah. So I think the only thing that we are encouraged about the Oxford and the Moderna trials is that the target that we're working with, raising antibodies against that target, protects against the disease. So we have this correlate. I think if you look isolated at the Oxford data and Astra, then it's quite disappointing that in the large vaccination group cohort only reached 64% efficacy at the peak response. Because that is bound. That is as good as it gets. It never gets better than in the peak response. Then it's going to fall, and it will likely fall maybe to 30, maybe 20. And then if you have half of the people accepting to take a vaccine, then you're not going to control the pandemic. So we really need effective vaccines here that are longer lasting than what we believe we will see from adenovirus.
Thank you. So I just have one final question for you, and it's a little bit perspective or taking us perspective on this huge collaborative and fast tracking initiatives that we've seen with coronavirus. So I think you touched a little bit upon it, Ali, but for the next great big pandemic we will encounter in the future, I think now we cannot say that will not happen. It will for sure happen. What are some of the benefits or the learning points we've had from this vaccine development that we can take to the next one? Maybe Ali, if you'll start.
Yeah, I will start because this is so exciting time for us developing vaccines. So as I said, we've been in the business for a long time, and we have seen all the big pharma lose interest in vaccines because it takes a long time and it's costly, so there's no big pharma developing vaccines. And we all saw that when COVID the pandemic started, it was Moderna and BioNTech and Oxford and University and AdaptVac. So all these small things, companies emerging with their technology. So I think, but on the other side of this pandemic, we will see big pharma being interested in collaborating on vaccines. And the most exciting thing is that we are all working with the same target. And now we are comparing all platforms. I think there are around 250 vaccines in pipeline on different platforms, and so we will come out on the other side and see how do they perform, how good are they at using a cellular response, and how good are the different platforms in using an antibody response, how long lasting are the responses. So it will enable us to make really good vaccines against other diseases like HIV and malaria and tuberculosis, because we will get this huge information package.
Yeah, thank you. And Max, you've probably also learned something. You already mentioned you were working on other vaccines. So
the successful proof of concept of this virus-like particle technology is key to us going forward, of course, as a small company. So as I said, we're really looking forward to human data on the system. And once that's done, we know we can handle. We've worked on the process for making the VLP and the antigen. We know we can scale this. I also know that we can improve on this. And so really it just emphasized that we can handle this and then some.
That sounds amazing. And I definitely think all of us share your excitement, looking forward to the clinical trials and this whole vaccine development journey overall, because it is really exciting times. So I would like to thank you all very much for coming today, and wish you all the best, and we'll keep an eye out for you and other vaccines and how successful they are, because I think we're all eager to go back to our normal days, normal lives. So thank you very much again for coming, and with this I'd like to say thank you all for joining, and this concludes our science talk for today.